Sir Arun Kedia is the Managing Director of Valve Life Sciences and Valve Lipids. Is a distinguished chemical engineer and visionary entrepreneur with over 3 decades of experience in the life sciences and pharmaceutical industries. He is widely recognized for pioneering advancements in lipid manufacturing and nanotechnology-based delivery systems. Founded Valve Life Sciences in 2003, later Valve Lipids in 2009, transforming them from modest ventures into globally respected innovators in phospholipids and lipid nanotechnology ingredients. Under his leadership, Valve Lipids has emerged as a trusted global supplier of high purity lipids for pharmaceutical formulations, nutrition, cosmetics, and advanced lipid nanoparticles. During the COVID-19 pandemic, Valve Lipids played a crucial role as an Indian contributor of specialized lipids used in global vaccine delivery systems, supporting mRNA vaccine development and manufacturing at a time when the world faced unprecedented challenges. Through a commitment to scientific innovation, quality, and collaboration, Mr. Kedia has positioned VAV as a pioneer in lipid science, bridging India's manufacturing excellence with the global need for advanced lipid technologies. VAV is an Indian pioneer of natural and synthetic phospholipids and the first to make the revolutionary egg oil oleova globally. So I invite Mr. Arun Kedia for his talk.
So thanks, Dr. Thakur, for the introduction, and thanks to IIT for providing this opportunity. So I'll try to give an overview of lipids. As you know, the subject is vast, and we'll have speakers who will talk more into detail about particular aspects. So my idea is to give give an overview of the subject. So let's dive right in. So what are phospholipids? I mean, you've heard this over and over again. Part of every living cell, which is part of every membrane. So what are the key properties? Again, trying to give an overview. So amphiphilicity because of the structure, self-assembly first into the bilayer and then into spherical structures, liposomes, whether unilamellar or multilamellar, and the ability to act as surface active agents because of the polar nature. So where do lipids come from? So we have natural sources of lipids and some common plant sources are listed. This is not restrictive. This list represents more what is commercially relevant, but virtually every plant source can have a lipid, again because they have living cells or membranes. Similarly, diverse range of animal sources. The most common commercial ones are egg, milk, marine. And then we have a range of synthetic lipids. Again, these could be also classified into semi-synthetic, where you are taking a natural molecule and then doing some partial modification, such as saturation or head group exchange. Or you could start with a completely synthetic chemistry approach. Then the applications depend partially on the source and partially on the nature of the lipid from that source. So the applications are sort of broad classifications, not supposed to be exact, but broadly you will have plant sources going into the listed areas, and animal lipids are particularly preferred for injectable or parenteral applications because of the specific composition. And I can highlight that the absence of phosphatidylinositol and phosphatidic acid, or let's say reduced phosphatidic acid, and the absence of inositol makes it particularly preferable for parenteral applications. Parenterals can also apply to intraocular, or pulmonary, so anything which is really parenteral. And then the synthetic lipids are classically applied into nanoparticles. I would like to highlight some of the significance of the synthetic lipids, why synthetic lipids are preferred. So they could have sharp phase transition or glass transition temperatures, which means much more control on the release profile. Then they have targeting properties, particularly if you want to attach ligand, so ligand mediated targeting, then it's normally only possible with the synthetic lipids. And then of course, because of the single molecular structure, you have very, very narrow polydispersity. So, I mean, this is a little bit amusing to me, but nevertheless, so there are four classical major phospholipids: PC, PE, PA, PI, and then some minor phospholipids. But here, why do I say amusing? Because, taking a classical soya lecithin, we've done a research project with the University of Utrecht in Netherlands to make an attempt to identify the lipid classes. And when they reached with a soya lecithin 1200 identified compounds, we said stop, please. So they said potentially it could be 5000 different lipid species. So, what I'm trying to highlight is that lipids is really complex chemistry. Natural lipids is even more complex. And understanding this complexity helps us to deploy them in a commercially successful way. When this understanding of the complexity of natural lipids is inadequate, then conclusions like they are unstable, they are difficult to predict, difficult to manufacture, difficult to characterize, encapsulation efficiency, all these problems stem from the inadequate understanding of the complexity of natural lipids. Once those are understood, there is far better control on the applications. Typically, serine, sphingomyelin, these are more in the animal lipids space. So, why are they key players in tackling drug delivery barriers? The drug bioefficacy depends on solubility, permeability, and target site uptake. Now, let's say, especially at an undergrad pharmacy level, people talk about bioavailability as just the reaching of blood plasma concentration. We have a private joke in VAV that if I just push the drug from one door to the other side, which means from the GI tract into the blood plasma concentration, PA bioavailability ho gaya. But does it reach the target site? Does it give you the desired efficacy? Questions sometimes which people are afraid to ask. So just measure the blood plasma concentration and claim efficacy or claim bioavailability. The patient or the consumer of the drug cares two hoots about this blood plasma concentration. They want therapy. So the ability to reach the target site is also critically important. If the drug is solubility limiting, then solving for solubility is enough. If it's permeability limiting, and soluble drugs also can be permeability limiting, or even if you solved for solubility, you could still have a permeability challenge. That's the next step. And then, of course, the third is the target site uptake. The target site can be an organ, a membrane, a tissue, or even a single cell. And I like to quote this example that if your target happens to be breast cancer, then inside the same organ, you have billions of cells. You have the cancer cells adjoining the innocent healthy cells, and if you are not able to target your drug to the cancer cells alone, then you are not an efficient, your system of drug delivery is not efficient. So your targeting should be able to target even single cells, and that's the test of a drug delivery system. How phospholipids overcome these challenges? They can solve for everything. The permeability is by bioadhesiveness. The absorption, you can measure bioavailability by whatever method you want to measure. And then you have the ability to target the drug to specific sites, and that, there are also multiple ways of targeting: active targeting, passive targeting, ligand mediated. So you see on the picture is a classical example of a ligand mediated liposome. So again, probably all of you are aware what is liposome. So these are self-assembled lipid-based drug vesicles. They can deliver at or administer virtually any drug or biologic or therapeutic agent. The fabrication of the liposome, though, is beyond these simple statements. So, should they be unilamellar, multilamellar? What should be the size? What should be the charge? So, there is a lot of complexity to designing the liposome. We face the industry and customers. So, there is often a fallacy in the industry. A liposome is a liposome is a liposome. And one simple statement that every nanoparticle is an engineered nanoparticle. I like to give the example of a well tailored shirt. So the more measurements you take of the person, the better the tailored shirt is. So if you design a nanoparticle exactly knowing what your drug is, what the dosage is, what the route of administration is, what the target site is, what is the release profile desired, is it milliseconds, is it 10 years, and we have examples of 10-year releases. So if you, the more factors you understand, the better you design your nanoparticle. And whether it's liposomes or there are also cochleates, there are several nanoparticles. And even within the liposome space, you have multiple ways of designing the liposomes. Somebody asked a question on manufacturing and stability. So that's also very closely related. I'm sure Dr. Mahesh can talk for the full day on encapsulation efficiency of liposomes. But okay, some other advantages, they are extremely biocompatible and biodegradable. So they are in the human body as natural as water. So you have no, toxicity or other factors to worry about because of the substance itself. You only worry about the drug toxicity or efficacy or release, but nothing coming from the lipids themselves. Improved biodistribution, prolonged circulation time. Again, this is a design factor. Do you need circulation time of minutes? Do you need for 1 week? Again, I mean, it can be as good or bad or as long or short as you like. We have an early stage research project going on with a Swiss, let's say, company, early stage, and which is to administer a drug post heart attack and to prevent ischemia. Now, understand the time window that after the initiation of a heart attack to prevent ischemia. इसके बीच में time कितना मिलेगा? And those kind of releases are also possible, at least in hypothesis. Of course, it's yet to be tested on humans, but at least by hypothesis, it's possible. On the other hand, you can have long releases. We have a drug-eluting stunt, which is now patented by Abbott, and it's in the public space. You can look it up, which is release of the drug from the stent over the useful life of the stent, which is 10 years, which means 150 milligrams of drug released in picograms per heartbeat over 46 million heartbeats. So you divide 150 milligrams by 46 million and you have the math. So you can have reduced drug degradation, targeted delivery. You have, like I said, I mean, multiple ways to design a liposome by size, by lamellarity, by many factors. You can bypass the first pass metabolism if this is a critical factor, not out of fashion, and then you have some pegylation, or you can have also other molecules to bypass the metabolism, first pass metabolism. You have ligand targeted, which means you can target very specific sites, and then you have the traditional liposomes mostly used in personal care, nutrition, or some routine pharma. So phospholipids are used as excipients to enhance membrane permeation, bioavailability, and controlled or sustained or extended release, lymphatic absorption, reduced cytotoxicity. We have seen, for example, in some highly cytotoxic oncology molecules, 180x reduction in dosage for the same bioefficacy. So you can imagine that when you reduce the dose 180x, you don't reduce toxicity 180x, you eliminate toxicity, because you will have the LD50 logic that beyond a certain dose the drug ceases to be toxic. So those are good examples. And of course you have solubility, stability. Uh, Phospholipids are used as emulsifiers classically, as Dr. Pradeep mentioned, for topical emulsions, but also for parenteral emulsions. Usually, you tend to use plant lipids for oral and topical, and animal origin lipids for parenteral emulsions. These are applications which are widely marketed from maybe quarter of a century ago. So these are very classical. Lipids in vaccines made a big noise very recently. So particularly for the mRNA delivery. Here I want to add a point that probably if you've been following the press after the post-COVID era, then some of the lipids used in the lipid nanoparticles were proprietary lipid. Now there is a commercial reason why particular lipids were used for obviously commercial protection. They were patent protected. So the SM103, ALC0315. The company launched the LNPs using those particular lipids without adequate safety studies on those lipids. And if you have kept up with the press, you had a lot of bad press for vaccines, particularly Pfizer got a lot of bad press. Some of those vaccines were considered pro-inflammatory. Of course, the press exaggerates, so some deaths were caused by the lipids, so-called. But the point I'm highlighting is that the more natural lipids you use as a first choice, the safer you are by the toxicity question. The second question is then the second approach can be to use lipids which are well researched or where the adequate long-term safety data is available. In the Pfizer vaccine example, lipids were used because they were patent protected, so they could be exclusive to the company launching them, but without adequate long-term safety data. And then you can have some risks. So some of the other common examples, oncology, again, I'll have a slide later. There are several marketed formulations in oncology, started with doxil, but paclitaxel, docetaxel, etc. And several under research or in various stages of clinical trials. Cardiology, again, there are well-established drugs, both oral and injectable forms. So lipostabil, lipodissolve, those kind of. Diabetes is under early stage research, and the opportunity could be to encapsulate the protein to prevent it from degrading in the stomach environment. Hepatology, well established, is 20 plus years. So if you all are aware, there is a product from Sanofi called Essentiale, which is one of the top 5 products for Sanofi globally. And it's classically used for hepatology for both alcoholic and non-alcoholic fatty liver disease. Drug-eluting stents, I mentioned, Abbott has patented this using our lipids, and now it will enter human trials shortly. So just a glimpse of several products marketed, with lipids. So at least this list should break the myth that liposomes can be unstable. Working with lipids is complex. It's not really, but when you have globally launched products and some of them since several years, it's possible to do it as long as you understand the nature, complexity of the nanoparticle and the lipids. Something from our marketing team, of course, they have to sell. But beyond what is written on the slide, I want to highlight we are committed as a company to supporting Indian academic or non-academic research, and our lipids from 10 plus years, right from our inception, have been free for all Indian research. So please avail of this, let's say, facility. If your research work involves lipids, VAV is there to back you. Not only do we provide the lipids, we have a scientific team which is happy to guide your projects, safeguard or guardrail some of the common mistakes researchers or students make when working with lipids, and all of that ecosystem is available for Indian research. Yeah, then some we have been, of course, authoring. So when we started the journey, there was really no regulatory documents available. So there was no monographs, and we have been authoring some of the monographs. Our team has worked hard in the past. Now some of the monographs are published with the US Pharmacopoeia or the European Pharmacopoeia. Of course, the Indian Pharmacopoeia always lags by 10 plus 15 years. I don't know. Soon they will catch up. But and of course, there are also monographs which are open for comments. So we regularly contribute to the monographs, which bring standardization basically also to this science, because it's an evolving and emerging science globally. Also, our authors have contributed to the handbook of pharmaceutical excipients, those specific chapters which relate to lipids. Yeah. So happy to take questions.
Thank you so much. So we can take questions, couple of questions before we break for tea. Sir. You want this? Yeah, Mahesh.
Very nice talk. Thank you. So you mentioned that Pfizer had used certain lipids which they had manufactured and patented. So there are naturally available lipids that can be replaced with those and still get the same kind of encapsulation efficiency and all of that?
Okay. So, yes, first of all, absolutely, there are alternatives to those patented lipids. The patented lipids were not used because they are the best. There was no comparative study. They were used because of the patent protection. Okay, the Pfizer does not hold the patent because they licensed the technology from BioNTech. The encapsulation efficiency, sir, I would say depends on your art, which is the microfluidics. So it is not the choice of lipids primarily which will decide the encapsulation efficiency, and using your, let's say, classical unit operations, if I may use the term, you can go up to 95% encapsulation efficiency very easily. Microfluidics, for example. There are techniques which are available when you need to exceed 95, which is only in the case of cytotoxic drugs. Otherwise, they are not justified for biologics, but just for academic completion. Then you use something called active loading of the liposome. Practically, I mean, when we do internal discussions, I would say, active loading would apply perhaps to 1 in 5,000 cases where it is justified scientifically. Otherwise, for all the other cases, you can get excellent encapsulation efficiency using good microfluidics. But just to drive home also a point on this encapsulation efficiency, this is a very, very, very common fallacy. We see this also in very large companies, multinationals also. If they are not using microfluidics, it's also acceptable. If they use, for example, high shear homogenization. And, we will often hear this from customers, we are getting poor encapsulation efficiency. And then I ask, okay, what's the tip speed of your homogenizer? And then they look and what does it mean, tip speed? So how do you define a high speed homogenizer, high shear homogenizer? So is there a measure to the shear rate? Okay, if you don't know, then you are in the dark. So encapsulation efficiency depends on those factors rather than the choice of the lipid.
Yeah, one last question and then we'll break for tea.
Thank you, Mr. Kedia, for a very informative talk. My question is more from the initial part. You mentioned that lipids could be used for both BCS2 as well as BCS class for molecules. I bring in let's say the background more from drug discovery, often from preclinical to first-in-man studies when we try to check the druggability aspects. If the molecule is BCS4, normally the management would like to go back for structural modification because for BCS class for natural you do not have many options compared to BCS2. There are some penetration enhancers which may have some permeation issues on the intestinal permeability. If you think of complex formulations for liposomes, mixed micelles, they can't really load quite high amount of drug. And typically at phase 1, we do not know the dose. So can you throw some light if during the process of drug discovery, if you come across BCS class 4 molecules, are there any formulation avenues available for the CMC scientists so that they can still push forward for good molecules rather than going back to again drug discovery? Thank you.
So, first of all, very insightful question. My first comment is on a lighter note that the BCS classification is what we call the school classification. So, it's at least, I don't know, from my grandfather's time. Today, the more relevant questions would be, deeper questions. So, what is the dissolution? What is the pKa? Is there a charge effect? And a better characterization on what the drug delivery challenge is should come from something just beyond those four squares. So that's the first thing. We are quite convinced that even if it was broadly in that BCS class 4 space, you don't need to modify the drug. So very easily, depending on your route of administration, I assume you are talking about oral drugs, very easily these drug delivery challenges can be overcome. Now, what is the critical part? Of course, we, typically our colleagues will have a big questionnaire where they ask a lot of questions. Is the molecule vulnerable? Is it prone to oxidation, hydrolysis? Does it have a repulsion effect? What is the challenge of drug delivery or permeability coming from? And then understanding that you choose the right lipid, the right manufacturing technique, the right characterization. So, what kind of liposome I need? And we are quite convinced that you don't need to change the chemical molecule. So, and there are quite a lot of successful examples of development. We can share offline or talk about it. So, this is this is a very common industrial fallacy that if it is class 4, I should be paranoid, scared, petrified, ask someone to change the molecule. We don't believe that's true. Yeah. Again, just to give an example, I mean, if you're talking of BCS class 4 through the GI tract, how difficult that delivery challenge is compared to saying I want to deliver a drug to the back of the retina. And I can't obviously inject it in the eye. And if I put drops, it stays on the front. But these are well documented with the use of lipids now. So really complex delivery challenges can be solved with the use of lipids. But understanding lipids, understanding how you will make the nanoparticle, what kind of nanoparticle do I need? It is not loading limiting, because the first question is, if I increase bioavailability 200 times, how much loading do I need? So is it an HPAPI? Okay, if it's a paracetamol or a metformin where I need 750 mg, chances are you don't have a delivery challenge then with such drugs. So typically it will be, with very complex molecules, highly hydrophobic structures, or very low pKa or those kind of drugs, then the dosage required will not be mega. That's my...
Yeah. Thanks, Mr. Arun. So just a second. So I'll ask Mahesh to